All right, so this screencast is going to go through proteins, their structure, as well as their function. So our essential question is, how are the primary, secondary, tertiary, and quaternary structures of a protein different? And then how can proteins be destroyed? So biological functions of proteins, pretty much anything in your body is going to be done by a protein. Proteins have many important biological functions, everything from your enzymes, they're your catalysts, they make reactions happen faster by lowering the activation energy. Your antibodies, all of those defense proteins, anything that is going to keep you from getting sick, like you build up antibodies when you get vaccines or when you do get exposed to certain viruses and bacteria, those are all proteins. Proteins transport molecules, they move things, they regulate, they tell you what's going on inside and outside of the cell. They're there for structural support as well as movement. Our actin and myosin in our muscles are all proteins. So the protein building blocks are the amino acids. Hundreds are formed both naturally and synthetically. 20 of them are common in nature. Those are kind of the basic, 20 amino acids that we think of all proteins being made out of. So they're, essential amino acids. And they're alpha amino acids. The alpha means that the amine is adjacent to the carboxylate group, so it's just in the structure. You're going to have the amine before you have the carboxyl acetate group. So their structure, which I'm not going to make you learn the structure of the 20 amino acids like you would in an actual biochem class, but at physiologic pH, the amino acid has, so at a normal pH where, everything's in balance where it's supposed to be, it has a carboxyl group and it has an amino group. So you're going to have the amino group first, and then you're going to have a carboxyl group coming off the back. And there might be a side chain in our group. This is what makes each amino acid different. So all 20 amino acids are going to have a different R group coming off of them, but they're all going to have this amino group and they're all going to have this carboxyl group. So all the differences between amino acids, it just depends on their R groups. Some amino acids are hydrophobic, some are hydrophilic. The hydrophilic ones can be subdivided. They're polar. They're neutral, they have a high affinity for water, but are not ionic at pH 7. So just kind of some basic characteristics that you can have hydrophobic or hydrophilic amino acids. And then you can have negatively charged have ionized carboxyl groups in their side chains. So they can be positively charged, they can be negatively charged. And here's the positive ones. Positively charged are basic, on their side chain. So you've just got, they'll react with water to release the hydroxide anion. So a little bit of different characteristics, you don't have to worry about those. Here are the 20 essential amino acids in alphabetical order. If you, in biology, you know that your DNA is read 3 bases at a time, and actually it's your messenger RNA is read 3 bases at a time by transfer RNA, or by ribosomal RNA. And then transfer RNA brings in an amino acid to match those 3 codons. And each amino acid that comes in gets put in order of the protein chain. So these are the amino acids that all of your proteins are made out of, coded for by your DNA. And here's some of the structure of them. Some of the hydrophobic ones. And you can see they all have this basic structure with a different R chain or a different R group coming off of them. And then here's the polar neutral amino acids. So you don't have to know the difference between them, but they all have different chains coming off of them. Negatively charged amino acids, positively charged amino acids. And the important thing is these amino acids are held together by a peptide bond. So the linkage that's going to hold all of these amino acids together right here is called a peptide bond. And you can put them together, or you can break them apart. You can do the hydrolysis, which is breaking them apart, or you can do dehydration, which is putting them together. And it's just a matter of gaining or losing water during that process. So the basic structure of proteins is what I'm the most concerned about, is just making sure that you guys understand the different sequencing. The primary structure is just the amino acid sequence. So you literally would just write, right here. There's glycine, there's phenylalanine, another glycine, an alanine, a leucine, a serine. So that's just going to be the order of the amino acids when you just first write the letters out that they're going to be. Each protein has a different primary structure because they all have different orders of amino acids. The secondary structure is when they kind of start to coil and bend a little bit into these regularly repeating structures. And there's different ways they can fold into these sheets, or they can start folding into these circular structures with their R group sticking on the outside. But in the secondary structure, it starts to coil and bend. And there's different ways that this can happen. On a side note, when you get a perm, this is what makes your hair curly, is you change the alignment of the amino acids. You change the alignment of those proteins, and it makes the hair a different shape, makes them align differently. The tertiary structure is the final shape that it's going to take when it's functional. So it's kind of like the primary is just the order. The secondary is when it starts to coil and bend, like the stuff that likes each other moves towards each other. The stuff that doesn't, like the polar and the non-polar ones, start to move away or move closer to each other. The tertiary structure, though, is its final shape in water. This is the full three-dimensional structure of the entire peptide, the entire protein. It defines the function. It is when the protein becomes functional. So here would be, the final tertiary structure. So if it becomes functional at tertiary, what the heck is a quaternary structure? Well, that is when sometimes you have to have multiple tertiary structures get together to become a fully functional protein. So the quaternary structure is when you have subunits or individual proteins that form a larger protein. They group together. An example of this is hemoglobin. For hemoglobin, you actually have 4 proteins that are all linked together, making one functional hemoglobin. So here's the primary structure. Here's the secondary structure, starts to coil and bend. Tertiary structure becomes functional. And then here is a quaternary structure where there's 1, 2, 3, 4 different proteins that have to work together to become one. Not all proteins reach this quaternary structure, but some do. So proteins requiring prosthetic groups, we don't really need to worry about this, but it's just stuff coming off of it. I'm not even going to take you through it. So protein functions follow the shape. Fibrous proteins, they tend to have mechanical strength. They're structural components for movement. We find these in muscles, the actin and myosin. Globular proteins, so the ones that look like blobs, transport of things, regulation, so they, trigger things to happen in and out of the cell. Enzymes make reactions happen. So myoglobin and oxygen storage, as I had said, hemoglobin is an oxygen transport protein in higher animals. Myoglobin is the oxygen storage protein of the skeletal muscle. So oxygen must be transferred from hemoglobin to myoglobin because myoglobin has a stronger attraction than hemoglobin does. So it's just going to steal the oxygen from it. And the heme group is an essential component of the proteins hemoglobin and myoglobin. And that's what you need iron for. Iron, the iron 2 ion in the heme group is the oxygen binding site. So you've got to have iron to allow the oxygen to bind, and each hemoglobin contains a heme group, which can hold 1 molecule of oxygen. There are variants to hemoglobin, which, like sickle cell anemia, that's what this is showing. Hemoglobin is shaped incorrectly. A fetus has a unique type of hemoglobin with slightly greater affinity for oxygen than typical hemoglobin. So when you are in utero, your hemoglobin is more attracted to oxygen. Another variant is the sickle cell hemoglobin that causes red blood cells to appear altered. So they look a little bit different, and they can cause problems with them getting clogged or forming clots. It can decrease the life expectancy of somebody that has sickle cell anemia. So we have proteins in our blood. They can measure how much protein is in your blood. All the different classes within your blood plasma, they can all be separated out into different types of proteins. Most of the remaining plasma proteins synthesized in the liver. So after you've got all these other types, the fibrins, the globulins, all of these alpha through gamma proteins that you have, the rest of them are going to be made in the liver. And a hallmark of liver disease is reduced amounts of one or more of the plasma proteins. So if they're trying to check your liver function, they will measure your plasma proteins. Whenever you are having your blood drawn and they are checking your liver function, like it'll say ALT or AST on the piece of paper, it's measuring how much of these proteins you have present. So how can a protein be destroyed? That's called denaturation of proteins. So you're going to denature it, meaning destroy it. Extremes of temperature and pH have drastic effect on protein conformation or on the configurations. It causes them to fold incorrectly. It causes them to unfold when you heat it, and then they refold back incorrectly, and then they don't work anymore. This is why very high fevers are dangerous, because it can denature the proteins in your brain. It doesn't alter the primary structure, but it does alter the secondary and tertiary structure of the protein. Because structure in the 3D shape matters in the activity, proteins are very susceptible to changes in those conditions. So you can end up with breaking down into smaller peptides so you can break them apart. That's hydrolysis. Changes in pH will do that. Sometimes enzymes will do it. Like you'll have enzymes that'll break apart your proteins. This is how you digest food, but you just don't want to digest your own proteins, as well as temperature. And then breaking up the 3D shape, temperature, heavy metals, detergents, mechanical stress, organic solvents, all of those things can destroy a protein. So the effect of temperature, because that's primarily the one that we think of as causing the biggest problems for us. As the temperature increases, the first effect is to increase the rate of molecular movement, so it makes the molecules move faster. The bonds within the proteins vibrate more violently, and eventually the weaker hydrogen bonds, if the temperature gets high enough, and the hydrophobic interactions are disruptive, meaning you break those bonds. And then you lose the three-dimensional conformation or the three-dimensional organization, and the protein becomes disorganized. You can also do this with pH. When the pH of a solution containing a protein is changed drastically, the acid or base will change the charge of the protein, and that causes big problems with the way that it's functioning or its structure. It interferes with salt bridges and hydrogen bonds that stabilize the tertiary structure, so then the whole thing can fall apart. So these other things like mercury and lead, that's why they're so dangerous. They cause problems with our proteins. Proteins have several dietary purposes. We break them down for energy. The amino acids we can then use to make our own proteins. That's what biosynthesis is. That's why they say you are what you eat. And then the amino acids which cannot be made in the body are essential amino acids. You have to get those from your diet in order to make your proteins. So these are the essential amino acids, meaning you have to eat these. These are the ones that your body can synthesize on its own. So that concludes our discussion for proteins.